Hello everyone and welcome to today's video. So today we're going to be going over the lab for the muscle models we have in the classroom. So we have this model right here which is showing one muscle cell or muscle fiber. So we're going to talk about the small components inside there called the myofibrils and the different bands involved and so forth. So I'm just turning this video around to imagine you holding this in your hand. Then the other model we have here is slightly different because I like this one because it emphasizes another part of the muscle fiber, and that's the sarcoplasmic reticulum where calcium is stored, and these triads, which are the T tubules in them, and they have the terminal cisternae on the side. So these are the two models we're going to go over. So first, starting with this one here, let's find a good little stopping point here. So there's looking down on it. You can see all these little myofibrils running down through. So let's go right. Let's see, then right here is actually a pretty good stopping spot. Decent angle here. So let's start drawing our lines to what we want to label. So first one here is this connective tissue on, well, not connective tissue, but the cell membrane of the actual muscle cell. So that's one we want to focus on. Then we want to focus on the connective tissue around this muscle cell. So this is just showing one muscle cell. Note that these cells are then grouped together into fascicles, which are surrounded by a connective tissue called the perimysium. And then all those fascicles are then surrounded by another connective tissue called the epimysium. So we're only focused on one of the mysiums here, and I'll write that after we label. All right, I can write them now, why not? So this one here, this is called the sarcolemma. And then this is called the endomysium. So the connective tissue layer right here, and then this one is the cell membrane or the sarcolemma. So then coming up through here, what do we have coming down here? We're not going to go into the intricacies of a neuron, but this is the motor neuron. So this is the myelin layer on that motor neuron, but we'll just label this as the motor neuron for now. So motor neuron right there, and then we reach the end of the motor neuron, which is let's draw this one a little further out, which is the axon terminal. And this whole region up here, this whole region in here is called the motor endplate. So the motor endplate, you have endplate depolarization that happens there. So the synapse, well, the action potential travels down the neuron, hits this motor endplate, and then here is where we have I'm not going to label it, but these are the synaptic clefts. And then right here is where you would have all the acetylcholine receptors lining this region to cause a depolarization event to go inside that muscle cell. Over here you can see these layers of myelin. So axons are myelinated. This is a white layer on side, and this right here is a cell body. Not a cell body, but a nucleus of a Schwann cell. But some of those things will be saved for the nervous system, but just highlighting what some of these components are up here on the neuron coming in. But the key part is the axon travels, the axon is in the middle, and the action potential travels down this axon to the motor end plate and branches into the different, so this would be one motor unit as it comes down. So now what do we find inside the muscle cell here? I believe on this model, these things right here are the mitochondria. I'm not going to label those, but then down here, we have the nuclei. So this is a nucleus right here. Now remember, skeletal muscle can be multinucleate. So when I move this model around, we'll see some bulges out here with like a little bit of purple underneath, and those are other nuclei. So that's why I'm thinking these organelles right here in this model are the mitochondria. Next thing we have in this model, it doesn't look great, but it's just like this granule looking pigment right here. So I'll just label it to this one right here. On this model, this one is the Sarcoplasmic Reticulum. The Sarcoplasmic Reticulum, or SR. Now this is the same thing as the smooth endoplasmic reticulum in the normal anatomy of a cell, so that's where calcium storage is. And again, over here on this model, we'll see it much better. And so it's like I said, it's not a good description on this model here, but this one is a good one for the microscopic anatomy of the cell down here, because now when we get inside, this is when we get to the unique thing. So this is where we have the different bands that occur in the myofibril. So all these right here, we'll just highlight them all. Oops, I didn't want to erase that. These individual things are myofibrils. Myofibrils. So each individual unit right here, it's a myofibril. Now, what are these little lines on it? So hard to see on this one, but I'm going to try to name them. So the dark pigmented areas, these are the A bands. So if I were to draw it, let's go from, I could do this end one right here. So that one and that one, that whole region there, I'll name. So that is the A band. So that is where we find the thick filaments. All right, and next up here, we have the M line. So the M line is that little black line we see in the middle of these dark pigmented areas. Remember, the dark areas are the A bands. So this one here is the M line. Now, we have these light bands on the outside here. So these light bands, draw another line in here, kind of running out a little bit of space. But these clear lines, not clear, but these little pink lines right here. Actually, let's draw one to this one up there. So these are the I zones or I bands. And they are the light bands where we have the thin filaments only. So here, I bands. And then within that I band, again, running out of space here, we'll draw it down here. We have that line going all the way up through the middle here. So this line in the middle here, the line in the middle here, those are the Z discs. And then so Z disc to Z discs then is considered one sarcomere. So here, there to there, we'll draw this line coming out here. That is considered one sarcomere. So that's one contractile unit in a muscle cell. So when Z discs move closer together, that's when contraction is happening. So that's everything that I think is important on this model here. Again, the A bands, the M line, the I bands, and so forth are a little harder to see on this model. There is a representation of these on the next model. I won't draw them all out, but I will highlight them and show where they are. So let's clear the screen here. So I hope you can take a screenshot of this if you are using it. And let's move on to the next part of the models. So moving on through here. Oh, I wanted to show these. So up here you can see the other nuclei that are sitting in a cell, that dark pigment, purple. And here you can see straight down on a cross section of it, all these myofibrils and how they're packed together in here. And here is where you can see how the T tubules go down in. So we're kind of going to show this on the next model here. But so calcium release has to come through all these regions so that we can get all these units contracting together. So here, this model, it doesn't focus on the T tubules coming in. And that's why this other model here is important for this. So here, that's this model. So just going over a few important parts here. The yellow, this is the SR. So the yellow is the SR right here. I won't label everything, but the sarcoplasmic reticulum. The red blobs here are the nuclei. I believe these ones in here are the mitochondria. But nuclei, mitochondria, doesn't matter. They are all there. So here, this sarcoplasmic reticulum extends through here. Over here is actually the representation of the thick and thin filament. So right here is the Z disc. So we can see that little Z line right there. So then right here then is the I band. Then the thick filament will go from here to here. So that would be the A zone. And then right here would be the M line. So right there is the M line of the A band, the Z disc, and the I band are where this overlap occurs here. So again, just another representation where we find this. But right here we have the T tubules as well. So right here, each of these are called terminal cisternae. And then where the T tubule juts down inside, so if you would look on the side here, there'd be a little hole showing, so this is the sarcolemma right here, and then it goes into the cell. So that this action potential can travel through the cell, and we get a release of calcium throughout the entire cell. So this travels down through there. So there's one terminal cisternae, there's another terminal cisternae, and then you have the T-tubule in the middle, and all those together are called the triad. So triad, three things, two terminal cisternae, and a T-tubule going down the middle. So that was highlighted well here on this model, and that's why I wanted to show it. And there's also another representation showing the different banding structures we find in a muscle cell on a model. So if you have any questions on this, feel free to reach out to me, send me a message, and I'll get back to you. But other than that, this is all I have for this model video, and I will continue to play it here as we go through. Hold on, let me erase. Let's play it through here as it shows the rest of this model. But yes, that is all I have for today. So if you have any questions, feel free to reach out and I'll get a hold of them. Oh, here you can see the different fibrils in there. Again, it's not great there. You can see them a little bit too. This is the side of the model we focused on here. But that is all I have today. Hope you all have a great day, and I'll see you all next time. Bye-bye.